First, we're talking about mitosis. And this is your average. Now, the first step of mitosis is interphase. Now, in interphase, centrosomes appear. The second stage of mitosis is prophase. In this stage, the centromeres begin to go to opposite ends of the cell. Now, the nuclear membrane, nucleoli, disappears, and the genetic material comes together to form. Also, the nuclear membrane begins to. The third stage of mitosis is prophase. In this stage, the centromeres have completely moved to opposite ends of, while at the same time, the chromosomes in the middle have condensed even more and have formed kinetochores at their centers between the two chromosomes. Also during this time, the nuclear membrane has completely fragmented. Now, since it is completely fragmented, it allows for the microtubules between the two centromeres to invade the nuclear envelope and attach to the kinetochores in the chromosomes. The fourth phase of mitosis is metaphase. In this phase, the two centrosomes are now complete opposite poles of the cell. Now, while this is going on, the chromosomes have lined up in the middle of the cell or the metaphase. Now, microtubules from both of the centrosomes attach to the kinetochores of each of them. The fifth step of mitosis is, this is the shortest step of mitosis, and it begins with the chromosomes being cleaved and each being pulled toward the centrosome centromere. Now, while this is taking place, at the same time, the entire cell elongates. By the end of anaphase, each side of the cell has its own complete set of chromosomes. The sixth and final step of mitosis is telophase and cytokinesis. In this step, the chromosomes have now moved, or are now at completely opposite ends of the cell. And on the opposite ends of the cell, the nuclear membrane begins to form on each side with fragments of the original nuclear membrane. In the nuclear envelope, chromosomes that were there have become less condensed, and nucleoli have reappeared. Also, the centrosomes that were at opposite ends of the cells and pulled the chromosomes to each side still exist, but are no longer. Telophase ends with the beginning of the division of the cell membrane. And then cytokinesis begins with the complete division and the forming of two new daughter cells. Now, why this happens, the nuclear membrane of each cell is completely formed, and the chromosomes that have begun to become less dense are now, once again, genetic material just floating freely around the nucleus as in.
Okay, now we're going to talk about meiosis. This is meiosis 1, and the first step in meiosis 1 is prophase. In prophase 1, the nuclear envelope begins to deteriorate, and the homologous chromosomes within pair up through synapsis, where they bind together through protein structures. And during synapsis, crossing over takes place where compatible and corresponding genetic material from each homologous chromosome switch. And next, in prophase 1, the centrosomes begin to move apart to each pole, and mitotic spindle fibers are formed between each. Finally, in prophase, the spindle fibers connect to the kinetochores on each homologous chromosome, and the homologous chromosomes will begin to move towards the middle of the cell on the metaphase. Alright, the next stage in meiosis 1 is metaphase. In this stage, the homologous chromosomes line up at the metaphase plate, which is in the middle of the cell, and the sister chromatids are connected to the centrosomes by kinetochore microtubules, one to each. Alright, the third step in meiosis 1 is anaphase 1. In this step, homologous chromosomes separate and begin to move towards opposite poles by the spindle apparatus. The spindles are connected to the centromeres of the chromosomes, and because the centromeres keep sister chromatids together, they both move towards the same. And the last stage of meiosis 1 is telophase 1 and cytokinesis. In this final stage, there's a haploid set of sister chromatids in each pole of the cell, and a cleavage furrow is formed in animal cells to begin separation of the cytoplasm. And once this cleavage furrow is formed, cytokinesis finishes separation of the cells into two haploid daughters. Right, the second step of meiosis is meiosis 2. The beginning of meiosis 2 starts with prophase. In prophase 2, centrosomes form and mitotic spindle fibers form in between. The late stage in prophase 2, the chromosomes will begin to line up on the metaphase 2 plate in the middle of the cell to further divide. The second step in meiosis 2 is metaphase. In this step, both daughter cells from meiosis 1 are attached to the microtubules at their kinetochores because they're already lined up at the metaphase 2. Right, the third step in meiosis 2 is anaphase. In this step, sister chromatids break apart because the centromeres that attached them before break down and they begin to move to opposite poles of the cell. The final stage of meiosis 2 is telophase 2 and cytokinesis. In this stage, the sister chromatids move to opposite poles and begin to decondense into genetic. This is the nuclear envelope reforming. And then a cleavage furrow occurs as seen before, and at the end, cytokinesis completely divides into two new cells.
In conclusion, mitosis creates two diploid daughter cells, which are each genetically identical to each other and the original parent cell, while meiosis creates four haploid daughter cells, which are each genetically different from the parent and from each other. Mitosis is important because it enables multicellular adults to arise from zygotes, and it also produces cells for growth, repair, and in some species asexual reproduction, while meiosis creates gametes and allows for genetic variability among gametes.